Yeah, sorry.
As usual, introduce
Yeah, exactly. So in CGSI tradition, I have to introduce myself. So I'm Ben Raphael. I'm a computer scientist, I guess, these days, computational biologist at Princeton. I'm glad to be here. I'm sorry that I was not here earlier. I guess I'm maybe I'm an organizer emeritus now I'm steering committee, which is sort of like, I used to be one of the organizers of CGSI, and this year I wasn't due to some prior commitments. But I believe very strongly in CGSI and the community, and I'm here all day today, all day tomorrow, and Saturday, and I'm really happy to talk to any of you and the students, and if you want to talk about research or careers or anything, I'm happy to do it, and I look forward to engaging with everyone in the next couple of days. So today I want to tell you really just one story about kind of one idea, but I think it sort of illustrates some of the importance of developing methods for new types of data. And this is about looking at metastasis in cancer. So my research office requires me to have that slide, so there it is. So
Can you go back?
you can ask me questions about that slide. So I know there's been a little bit on cancer. Jinx Shalinap gave a talk on Friday, which described some of the topics I'm going to talk about in terms of cancer. So cancer, as has been known for a long time, is an evolutionary process. It's a process of mutations accumulating in cells, somatic mutations mostly that develop after we're born. And one of the real goals in cancer research in the past decade or so, as DNA sequencing has become cheaper, is to really identify what are the mutations that are causing cancer and distinguishing those from the passenger mutations. And then in the past 2 or 3 years, as sequencing has become even more cost effective, we've been able to actually drill a little deeper and ask not only maybe what are the driver mutations that cause cancer and what are the passenger mutations, but really kind of dig into what exactly is this evolutionary process, and can we infer this evolutionary process of mutations accumulating this population of cells from various types of DNA sequencing data. And there's lots of reasons why we might like to do this. We might try to find mutations that are responsible for drug resistance, or even identify mutations that are selected within a population. But the reason I want to talk about is actually to look at one of the really most important from a health perspective properties of cancer, and that's that cancer cells metastasize. So metastasis is a process where cancerous cells leave the site of the primary tumor and travel through the bloodstream or through the lymphatic system. Cancerous cells traveling through the bloodstream and lymphatic systems to initiate new tumors at distant anatomical locations. And it's really essential to understand this process. Metastasis is really the killer in cancer. Most patients, die because of metastasis, not because of the primary tumor. And so, we'd really like to understand what exactly are the drivers of metastasis. Are there mutations that give cells the ability to metastasize, or, other types of changes, epigenetic changes, gene expression changes, et cetera? How does understanding this process perhaps help us think about how we might intervene in cancer? And in particular, there's sort of a paradigm of whether or not you should remove the lymph nodes, these sort of locations here, the lymph nodes surrounding your primary tumor. And if metastasis first goes to the lymph nodes before it goes elsewhere, then it might be a good idea to surgically remove those lymph nodes at the same time you perform surgery on the tumor. So what exactly is this process? Okay, so there is a lot known, but there's a lot that's unknown. And what's now become sort of possible is to start to ask questions about this process by looking directly at the mutations. So the traditional view of metastasis is that each metastasis is the result of a single event. It's a single cell that leaves the primary tumor at some time, travels through the blood or the lymphatic system and seeds the tumor, which then acquires new mutations as it goes on. And maybe a cell leaves this metastasis and founds a new one. So each metastasis is a result of a single event, or a so-called monoclonal formation of a metastasis. But now we could just start to ask this question perhaps directly, because if we're able to, and in some cases we are able to from a patient, sequence the primary tumor and one or more metastases from that patient. Then we can go measure the mutations, here sort of measured represented abstractly, in each of the samples. So I have a primary tumor, 2 metastases, and I indicated for each of these somatic mutations whether it's present or absent in each of my samples. And then we could make use of the decades of work that evolutionary biologists and computational biologists have undertaken to build phylogenetic trees that represent these samples. So here in the cartoon version, I've shown you this tree with 3 samples, and the tree that sort of explains this data in the minimal way or the most likely way if we have some probabilistic model of evolution. And what I've shown here on the edges are where I inferred that each of these mutations occurred. Okay? So now we can start to see interesting things. Like we might see that, look, in terms of the mutations, these 2 metastases are more related to each other than they are to the primary tumor. And maybe that tells us something about how these metastases formed and migrated. Yes?
So question, do you think specifically of these mutations as mutations related to cancer, or you would just treat them as markers? Because then you could look at many other markers like STRs, which mutate more quickly.
Definitely, yeah. So I guess for the purpose of this talk, we think about somatic mutations, but not necessarily cancer-causing mutations. We're happy to have any somatic mutations, because all we really need are markers of cell division. Yeah, so STRs, any various types of mutation data could be used. Okay, so so now we can try to infer the past, right? Just using phylogenetic techniques. And this has been a very hot topic in cancer research in the past couple of years because we've started to have access to the data to ask these questions. So here's just a few of many, many examples. Colorectal cancer patient. There were 2 liver metastases here in red, 5 pieces of the primary tumor that they sequenced independently. Here's the tree. Again, you can see the 2 liver mets are more closely related to each other than to other parts of the primary tumor. Here's another study in colorectal cancer, also in the lymph nodes, these sort of parts of the lymphatic system that are closest to the tumor, and sort of from these trees, the L's here are the lymph nodes, P's are sections of the primary tumor, M are liver mets, and you can start to perhaps make inferences about, did the tumor first travel to the lymph nodes before going and seeding distant mets? And in fact, the conclusion in this paper was that seemingly that didn't happen in a large fraction of the patients, that somehow in some cases, the liver mets, seem to be spinning off from the primary tumor without first going through these adjacent lymph nodes, which has implications for how you might do surgery, should you remove these lymph nodes or not. Okay, and here's just another example in breast cancer. Again, each tree from a single patient. We're looking at one individual patient, and we're trying to infer what happened in their tumor. Okay, so this is one type of analysis. Yeah.
So your tree is over the metastasis in the body.
Yeah. So we've taken, in this case, it was 2 different liver metastases from the same patient, 5 different parts of their primary colorectal tumor. And we measured the mutations in each one, and we built a tree out of them. I didn't tell you exactly how we built the tree, but we're going to come back to that. Okay? Yeah?
Maybe it's technical. How do you know the primary tumor?
Great question. We'll come back to that. Often we might know it because the oncologist or the pathologist told us what the primary tumor was, but not always. If you have a patient that comes to you and they just have cancer in their liver and cancer in their colon, which one was the primary? Great question. We'll come back to that. Yeah.
Could you sequence the blood and then say compare that DNA to all the other samples you got, and then try to assign the primary based on which ones are most similar? Or?
Sequence the blood. Well, the blood would give us, for the most part, the germline genome, unless you're thinking about tricky things like circulating tumor DNA. But that's a whole other topic we want. Yeah. Right. So we can. In fact, they have the germline. That's what actually this N is the normal. So this is sort of the germline genome of this individual that they obtained from the blood or from some adjacent tissues.
That's how you know the root of the tree?
That's how you know the root of the tree, yeah. Here they did neighbor joining, so they didn't really root the tree, but presumably you could kind of root it on the normal. Okay. Here there were 2 different normals, so there's kind of like blood and skin or something, and you can see that they weren't kind of identical, but close enough. Okay, good questions. Keep them coming as we go through. All right, but then other recent studies that dug into things started observing really kind of strange and curious results, and quite fascinating. So this was a study that was done in prostate cancer. Here, these 3 panels are 3 different prostate cancer patients. What they sort of saw in this first patient here is that cells from the primary tumor, they inferred, migrated, and formed these metastases sort of locally. And then in some second wave of migration, cells left those local mets and then went and formed tumors at other parts of the body. And then somehow just, I guess I'm recorded so I won't, just all hell broke loose, right? And there's just tumor cells going all over the place, back to where they'd been before and coming around and just, it's crazy, right? Tumor cells everywhere. Here's another example, prostate cancer, another patient. Again, cells going up here, seeding a tumor, then coming back here, and then over here and around, and just crazy stuff. So they sort of said that, about a bunch of the patients, they saw what they called polyclonal seeding. So there's not a met founded by a single migration, but by multiple migrations of cells to the same place. And in some cases, reseeding, so cells coming back to where they had been before. All right? Really changes how we think about metastasis. Another study shown in a slightly different way. So this is a breast cancer study, 1 patient. Here's the primary breast tumor. The sort of thickness of this tree is kind of representing the cell population. And every time there's a new color, that's like a new, tumor clone with a different mutation sort of growing out in the tumor. So from the primary tumor, you see this expansion of these different clones with different mutations, and then they find them in the lung, the brain mets, kidney mets, liver mets, rib, et cetera. And it's sort of a complicated picture, but if you kind of look at it in detail, so you say look at the liver and the kidney, what we see is there are multiple distinct clones with distinct mutations that are shared by both the kidney and the liver. So somehow neither of these could have been a single event. It had to be multiple distinct clones, which they called, in this example, multiclonal seeding from the primary to the mets. Okay, so now, from the early looks at DNA sequencing data, it suddenly seems that maybe we don't know so much about this process of metastasis. And this was a review from a couple of years ago that sort of raised a lot of these questions, right? Is it metastasis a monoclonal event, or is it polyclonal? Can you have cells from one metastasis seeding another, or are metastases always spinning off from the primary tumor? Can you have them going back to where they were before? All sorts of crazy stuff. And if you look at this review article, if you look at some of the literature, you'd see that there wasn't really a rigorous way proposed to address any of these questions. Sort of a kind of ad hoc process, certainly not up to kind of the CGI standards of computational rigor, right? So let's take a little deeper look and see some of the strangeness that might happen. So here was this one tree that I showed you. They built this, I believe, via maximum parsimony or neighbor joining or something, but here's the tree. We went back and looked at this data, and we took the mutations, and they built this tree from 412 single nucleotide changes. And when we assign these mutations to their positions on the tree, what we saw was that about 10% of the mutations occurred more than once on the tree. Okay, so this is a somatic mutation, not in the germline, somehow happening, say, once on this branch and independently on a different branch of the tree. And in fact, these 2 branches are quite interesting. The numbers here are telling us the number of such mutations that are elsewhere in the tree. If you see this branch, there are 20 mutations that occur on this branch of the tree, separating the met from this part of the primary. And those 20 of those 24 also occur here on this branch. Yeah.
What do you mean same mutations?
Exact same single nucleotide letter change in the DNA sequence.
Okay, same site in the
Exact same site in the genome.
These are all SNPs?
All SNPs. Single nucleotide mutations, somatic mutation.
Yeah.
No large scale stuff. No, same single letter change. And so you're asking all really good questions, because this seems like perhaps a little bit strange. Yeah. Sorry, was the tree built?
When the tree was built, was it a homoplasia where tree building happened?
Yeah, because it was maximum parsimony, so it tried to find the most parsimonious, the fewest changes, and somehow the fewest changes leads to these homoplasia events. So does even the requiring the fewest, yeah. Okay, so there's all good questions. The most questions I've gotten so far on this slide, because this all seems a little bit strange to people, it seems like. And it is a little bit weird, because homoplasia is like a really big deal in evolutionary biology. The canonical example is like maybe the evolution of wings. Birds have wings and bats have wings, but that doesn't mean that the trait from wings gives us something that can describe how bats and birds are related. In fact, wings evolved multiple times over the course of evolution. So this idea of, convergent evolution is a really big and important issue. So we'd say, isn't it strange that these same mutations, are these somehow such amazing mutations that they happen twice over the course of cancer evolution? Strange. Or maybe they're mutations that there's just a really high mutation rate in these positions, and so these positions of the genome mutate more than they should. Perhaps, but that also seems strange. And so, the conclusion is that sort of, at least if we pulled some off-the-shelf phylogeny algorithms, in this case it was maximum parsimony, we got sort of curious results. So we should perhaps think about this some more. And there's a big problem. Sorry?
Each node is a single cell?
Now you're pointing to the big problem. So what's the big problem here? The big problem is what are these nodes?
These are metastasis.
Yes, right. They are. So what did I do here? I took I cut out a piece of the liver met here, met1. I cut out a piece of it, and I ground it up, and I threw it on my DNA sequencer, as we usually do. All right. But the key point is that I ground it up. There were many cells in that sample, because we traditionally sequence many cells at a time. Only recently have we been able to try to sequence the DNA of single cells, but that's a whole other topic. But all of these were not homogeneous samples. Any time I cut out a piece of a tumor, there's a collection of cells that may not have the same mutations. Okay? So the issue here is that we built this tree that relates samples, but mutations accumulate in cells. Okay? So this is sort of an issue, and you can kind of read about this. David Posada, who's a evolutionary biologist, kind of wrote he's now interested in cancer evolution, and wrote a really nice review saying these multi-region tumor trees are not phylogenies, an issue that many in the cancer community weren't really quite aware of. And you can get weird results by doing this. And I think this is a sort of more general idea in computational biology that we should be aware of, which is, it's important that you use the right tool for the job, right? So biologists might have spent millions of dollars and many years constructing this beautiful data set, a data set that no one has ever seen before. It's beautifully decorated, it's amazing, and then it's time to analyze it, and so we just pull off some beautifully engineered Python or R package that's extremely well engineered, but they don't match up. It's not the right tool for the job, right? So part of what we should be doing in this community is building the right tools for the job and making sure that they're used appropriately. So what's the right tool for the job here? Well, the notion is, if we're going to do bulk sequencing, we could think that maybe we can do single cell, but for the moment, most of the data is bulk sequencing. So this is not actually our data, in that we don't really have the presence or absence of mutations in each sample. If we dig into the data a little more, what we really had for single nucleotide mutations is that we had all these reads from our Illumina sequencer that we aligned to the reference genome and decided to call the mutation as present or absent according to the number of reads that were there, and we used some Bayesian model or whatever to decide. But there's actually more information in the data than mutation present or not in each sample, because we could also, in fact, try to count the fraction of reads aligning to the position that tell me that there's a somatic mutation or not. This is so-called variant allele frequency. So if I see that maybe 40% of the reads at the position have the mutation, then that sort of suggests that maybe approximately 40% of the cells in that sample have the mutation. And if I sequence deeper coverage, this will become a better estimate. So our data is not digital, it's analog. Right? So this actually leads to sort of some interesting questions. Can we construct trees from analog data? Or sort of by analogy, species evolution has sort of all been premised on thinking about having the DNA of individual species. You measure the leaves of the tree and you try to find the tree that best explains that data. But that's not what we have. Instead, we've somehow measured mixtures of all the species on individual continents, represented at different frequencies. We have this analog data. So can we, from this mixture, where we don't even know the leaves, we have a mixture of unknown leaves, we don't know the proportions of each leaf in each sample, and we don't know the tree. It's a very underdetermined problem. But it's actually a very fun problem. And I hope, I don't know, I didn't see the video, Cenk talked about this problem. Cenk Shalinop talked on Friday. He didn't talk about this problem. If he had talked about this problem, he would tell you that he solved the problem. But in fact, many people have worked on this problem. So I'll say that we've also solved this problem. But it's actually a really interesting problem. Can you take this kind of mixed data and unmix it while simultaneously inferring the tree? It's a hugely underdetermined problem. So in order to have any hope of solving it, you have to impose some conditions on the evolutionary model. So the simple condition is actually to rule out homoplasy. I think this was essentially the question back there. If we assume that a mutation could never occur more than once, which is a reasonable assumption for single nucleotide mutations, then we can use that constraint and a bunch of other stuff to actually try to build these trees. Okay? So I'm not going to talk about that problem, although I'm happy to talk about it offline. I'm going to assume we solved this problem because people have worked on it for now a number of years. And there's a lot more, there's ... There's a lot more references here that go along with it. Okay, so we assume we'll solve this problem. Let's go back to metastasis to see if this is enough to tell us about migrations. Okay, so we've decided sequencing each sample and building a tree where we assume the samples are actually homogeneous is a loser. We'll see these fake homoplasy events. We won't really be able to quite infer what we'd like to infer. And so we need to instead use kind of specialized techniques that are aware of this mixture, and they essentially perhaps try to find the clones that make up this mixture and build the trees that are aware of these subpopulations of cells. Okay, so we attempt to do that. Let me just say it's not an easy problem, and different issues come up if you do try to do this with single nucleotide mutations. If you try to do this with copy number aberrations, which are very common in many solid tumors, it's actually much harder. But let's just assume that this problem's been solved. Or even better, let's just assume that we sequence single cells. And we sequence them, in fact, perfectly with no error. No one can yet do this. But we're just going to assume that that can be done or that these algorithms are perfect. So now I've given you this clone tree or this cell tree with zero error. And now we'd like to infer something about these migration events. Okay? You have the cell tree with perfectly. So we should be able to get the migrations, right? Well, almost. But in fact, yeah.
But you say the tree is the migration history.
The tree is the tree, and this is the key point. What's in this tree? This tree was built from mutations. Mutations are a marker, if they worked well and we have enough of them, for cell division. But they're not. There's another process of migration, right?
The same process.
Well, not quite. Cells migrating is not quite the same as them dividing. And they're, of course, related in some way. This one informs the other one. But, just to sort of say kind of abstractly, if you have perfect data, a cell divides into exactly 2 cells. So the cell division history is a tree. It has to be a tree. The migration process, though, if we think about migrations as represented between anatomical sites, does not have to be a tree. And this example shows that it's not a tree. Okay? So they're not quite the same. But of course, they're related. How exactly are they related? How are they related? And we have perfect data, right? This tree is exact. It's perfect. So what's missing? Why can't you tell me automatically what the migrations were?
The geography. this is the geography of your body.
Sure, geography.
I mean, where it was taken.
Ah, yes.
Geography.
We're missing, we know where the cells we measured were taken from, but we don't know where the ancestral cells were. Where was the ancestor of these two cells? Was it located in the red met or the blue met? That's not in the data. With perfect data, it's not there. We don't know the location of the ancestral cells. This is what we need, right? So we have to infer it. So how do we infer it? Well, we have to guess. We have to infer it, right? So let's just guess. Here's an example. I sort of guess. I have to choose a color for each ancestral node. If I choose the colors this way, then here's the migration history that I get represented here over this graph. So I've shown you each of the 3 anatomical sites, and there's a directed edge every time there's a migration. And this just corresponds to every time there's a color change in this tree. So I just guessed what the ancestral nodes were. I made the green here, I made the primary. Assume that we knew the primary. We'll come back to that in a second. And I guessed, what happened here. And I get 8 migrations, and I get cells going back and forth between, and I get crazy stuff, right? So maybe that was a bad guess. So maybe we should guess differently. If I guess, that all the ancestor cells were in the primary, now I get that there were 4 migrations. But I still get, somehow parallel migrations between the same sites. So could I do better than that? What does better even mean? What should we be trying to do? Now we have to think about it. It's an inference problem. It's not in the data. So we have to come up with some model. So perhaps one idea is that migrations, a cell leaving one site, migrating through the bloodstream, landing somewhere else, starting a new tumor, seems pretty unlikely. So we could assume that that's perhaps a rare event and perhaps doesn't happen that often. So maybe we should try to minimize the number of migrations. And that turns out to be a well-known problem. If I give you a tree, I just want to color the ancestral nodes to minimize the number of migrations. Yes?
But you could imagine a model with 3 migrations here instead of 4. If it's not single cells that migrate.
Ah, right.
Breed population with the 2 types.
Yeah, okay, so then, right, what actually sort of migrates? If we assume we know the cell division history and that all we're doing is labeling corresponds to a migration history, then I think in this example we can't label to get 3. We can't label the colors to get 3.
Population can bring migrating to a second location that you don't know about.
Oh, okay, well now all right, sure. If you start allowing that there's some site I haven't measured, then that's a new color that's not in my color set, and I'd have to be aware of that, yeah. So we could use that in the model too. We won't do that complication to start with.
The key question is whether you allow only single cell migrations or whether you consider that there might be a population of cells.
We're going to come back to that, yeah. So at the moment, we've done something very simple, which is all we're trying to do is just assign an anatomical site, a color to each ancestral cell, and do it in a way to minimize the number of migrations. So this is a well-known problem. This is actually just a maximum parsimony problem with a single phylogenetic character, and can be solved with classic algorithms, Sankoff algorithm, the Fitch algorithm. Have you guys seen these algorithms? Good. All right. So we can solve this. We can minimize the number of migrations. In fact, even back in the 80s, this idea of using the Fitch algorithm to minimize migrations between human populations was in the literature. Okay. So it's there. I just want to show you this quickly because it'll be important for the next thing we're going to do. How does this algorithm work? Well, it's very simple. We're just assigning a color to each ancestral node to minimize the number of migrations. If we want to do this, where we keep track of what's the minimum number of migrations for a subtree rooted at a particular vertex, if I assign the color C, okay? And we can compute that because it only depends, this best labeling of a particular node only depends on the color of the children. And so we just can percolate these measurements up. So just in this example, you can see right here at this node, if I label this node red, I would have 1 migration because red changed to blue. If I labeled it green, I have 2. If I labeled it blue, I have 1 migration. So that's the values of mu c at this node. Here at the leaf is very easy because it has to be the label of color. And then I could decide what's the minimum number of migrations at this node V by computing this recurrence relation looking at these two vectors of values, right? It's a very simple algorithm. And again, relies, this is the sort of optimal substructure property, right? All right, so this has also been used in one of the cancer papers from Saurabh Shah's group, who had a very beautiful data set for, unfortunately, a patient with very bad prognosis, because it was a ovarian cancer patient that had multiple mets all throughout the abdominal cavity, in the bowel, in the fallopian tubes, et cetera. They sequenced each of these samples. They built a clone tree, so they were sort of aware of the fact that each sample could be a mixture, and they used some fancy techniques to actually try to get this clone tree. And then they found a minimum migration labeling to infer a label for each of the ancestors. Okay? And now here's where this question about the primary comes in. Because in fact, in this example, and many of the other samples from this data set, there was a tumor in the left ovary and the right ovary and all throughout the abdominal cavity. So what was the primary? And in fact, one of the ways they inferred the primary was just by the minimum migration labeling. So in this case, the minimum labeling had the right ovary as the primary. And they had 13 migrations. And look at this. You have cells leaving, going to the small bowel in different migrations. Crazy stuff, crazy migrations. Yeah, but it's the minimum, 13. Yeah.
But it looks that there might be numerous minimum.
Oh my gosh, you guys are all so smart. You should be publishing papers in Nature Genetics, because there are multiple optimal solutions. And here's, in fact, all of them that all have 13 migrations. But the pattern is quite different. And in fact, even the primary tumor is different. Here it was the right ovary, here it was the left. Here there's crazy stuff between the small bowel, cells going back and forth between the left ovary and small bowel. Here not. Everything leaves the left ovary. There's some back and forth between the left and right ovary, et cetera. So what can we do now? How could we distinguish these? We can't distinguish them from minimum migrations. So in fact, at the moment, we can't say anything about what really happened. And so if you're talking to a cancer biologist and they say, I analyzed my data and there was reseeding in this patient, you'd say, really? How do you know there's reseeding? What exactly did you do to decide that there's reseeding? Because in many cases, you can't actually determine it. But this is a little bit unfortunate. So we're sort of modelers. So can we do some other things? Let's try to enrich our model for migrations. Can we distinguish these in some other way? Well, notice, they have these different structures of this graph. So let's try to use that structure. We could, in fact, also, one idea is that, certainly not every migration between anatomical sites should be necessarily the same. Maybe it's pretty rare that you go from, the ovary to the appendix, and it's much more likely that you go from the ovary to the small bowel. So you could think of assigning different weights to these edges based on what you think about the probabilities of migrations between those sites. Unfortunately, that would actually, you could weight them and use weighted parsimony. We don't really have good data to infer those rates yet because we haven't really seen enough migration patterns. So rather than do that, we'll actually look at structures, the graphs themselves, and try to score the graphs in a different way. Okay? And one observation, actually, I think this was your question, is, what about these kind of parallel edges? These parallel edges are kind of interesting. Maybe we should think about parallel edges. And there's actually a biological reason why parallel edges are interesting to consider, because there's some evidence that it's not single cells that migrate through the bloodstream, but clusters of cells. So you can imagine that a migration event can include many cells at once. And that would correspond to a parallel edge. So maybe we should count not edges, but count parallel edges in this graph. And we can name those co-migrations. So counting the number of parallel edges in the graph. There's other reasons we might like to infer parallel edges. Maybe we just sort of think that maybe there are two sites where at a certain point in the evolutionary process, one is very likely to spin off cells, the other one's very likely to receive cells. So we might get multiple migrations between the same site. But let's, for various reasons we could count these as different events. Yes?
But from what I understand, these trees are very incomplete. There might be lots of different cells that you didn't sequence.
Oh, here the data's, yeah, there is. In reality, yes. We're starting with just perfect data at the moment. But yeah, that'll come back in a second. That the tree, for now we assume the tree was perfect. Okay, and then the other thing we could do is we could start looking at the different graphs. And these migration graphs, which we derive directly from the labeling, have different features. And in particular, they could be, trees or directed acyclic graphs or directed graphs with cycles. They could have parallel edges or not. We'll call them monoclonal if all the edges are single, polyclonal if there's at least one parallel edge, sort of matching the language that's used in some of the cancer literature. And then we say that they're single source seeding if they're trees, multi-source seeding if you have these types of structures that give you DAGs, and reseeding if there's a directed edge. And this is the sort of essentially complete taxonomy of 6 possible structures. Okay? And now we've got 3 criteria we can use to start examining migration patterns. We can count migrations, migration number. We can count these co-migrations, or we can count for different patterns. And these are 3 different knobs that we can tune. We could try to minimize migrations without worrying about these other criteria. That's what the Sankoff algorithm was doing. Or we could try to somehow minimize co-migrations and not worry about migrations. Or we could try to say, I really want this migration pattern to be a tree, because I don't believe in reseeding and cycles and crazy stuff. So you tell me the minimum number of migrations if it's a tree. So these are 3 criteria. So now we have a much harder problem. It's not a single objective, it's multiple objectives. And in fact, there's sort of a trade-off between them. And there's not a unique optimum always. Depending on which of these knobs you care most about, you can get different things coming out. Okay, so just to show kind of one version of this. So here's, minimize the number of migrations first. As a secondary objective, if you have patterns with the same number of migrations, then secondarily minimize the number of migrations and require that there's a specific pattern. Okay? So now we can try to solve that down. How would we solve this problem? It's a different problem. How do we solve this? Okay, so given a clone tree, label each of the ancestral vertices to minimize the number of migrations. That was the first thing that we've already know how to do. That's Sankoff. That's easy. But now you want to minimize the number of parallel edges. And you want to require that after you've done coloring it, that graph is a tree. You want to rule out directed cycles, undirected cycles. No cycles. Has to be a tree.
You want to minimize or maximize co-migration? Because you're saying that might be a single event.
Sorry. Yeah, it's essentially kind of maximizing co-migration, minimizing number of parallel edges. Yeah, that's kind of weird terminology.
So
we count the.
Them to 1, when you
Yeah, exactly. So what we're counting, right? When we count co-migrations, what we count is we just count the total number of edges in this graph once we collapse parallel edges. We count each parallel edge as one event. Yeah.
So it's maximizing.
Yeah, exactly. So it's the most co-migration. Yeah. Thanks. That's probably a better usage of the terminology. Okay. So how do we solve this? Sankoff doesn't work because we lose this nice structure of just being able to look at, the direct children. Why? Because now we start having these global constraints. If I were to, decide to label this red because that's somehow the optimal thing to do here, and label this one green because that's the optimal thing to do here, then already I've created a non-tree structure in my migration graph. So it becomes a sort of global constraint of this labeling, and we can't do it with just dynamic programming in the simple way. We've sort of lost this optimal substructure property. So how can we do it? We can sort of brute force it in another way. We can use what's called integer linear programming. Integer linear programming is sort of like the deep learning of the 70s and 80s, in that if you didn't know how to solve your problem, you just wanted to brute force it, and you were working in the 70s and 80s, you would do an ILP, and there's good software to do it. Right, so we just did brute force because we don't want to think that hard. we just brute force it. And then just like deep learning, we have to figure out what exactly is the network, and is it convolutional, and how many layers, and all this sort of stuff. Here, what you have to do is you got to figure out how to turn your problem into a linear objective function with linear constraints. And just like in deep learning, where certain people are really good at this, but no one knows why they're good at it, they just are. There are certain people that are just really good at taking any problem and turning it into an interlinear program. And they're just really good at it. I'm unfortunately not one of these people. One of my former postdocs, Mohammed Al-Kabir, was. And so he could do this. And the first step is easy, actually. I could very easily tell you how to define this part. I could do. I could say, well, I need to count for each edge here whether or not there was a change in color. So I can encode with a 0 or 1 if there was a change in color. And then my number of migrations is just summing up over all the edges, these 0, 1 variables. So I have integer variables, and I have a linear constraint, so that part's easy. But then how do I do the rest of this stuff? Enforcing the tree structure and the graph and all that in an integer way, and it turns out you can do it. And I'll skip all that, but you can do it. So what happens? So now, here was this ovarian data. These all had 13 migrations. But now we can count co-migrations. This one has 10, the one that was published. You can find other solutions that have 7, right? Because they have more parallel edges, so you might prefer them. But also you could ask, well, I don't believe in any of this crazy cycles. So how many migrations would it take to get me a tree? And it takes 14. Only one more than the minimum. So now what do you believe? That it was 13 and that it was crazy, or that it was 14 and it was a tree? And on one patient you can't quite decide this, but if you start doing this analysis across many different patients, we can start to actually explore the trade-offs in these different parameters and start to sort of get a sense of this. Okay, very quickly, the question was asked, well, we did everything this way. We assumed this was right. The clone tree was perfect. And then we were just trying to label the anatomical sites to get the migration graph. But this is not going to be perfect. Could we use constraints on migrations to help us resolve ambiguities here? And we can do this. And there's actually a lot to do here. We've only sort of scratched the surface of this. So some of the ambiguities that we could attempt to resolve are sometimes when we build the tree, instead of getting one cell divides into 2, it looks like there's a multifurcation. This is some loss of resolution in our data. So those are not good. If you actually infer migrations and there's this multifurcation, you'll require more migrations, but it's sort of an artifact. And then, this data, and I didn't really go into this, but this whole idea of getting the mixture, there's all this ambiguity in what the analog frequencies are, and you don't get the tree right. And so we could actually try to simultaneously build the tree and infer the migration scenario. So, in this case, I guess this example, when I had the multifurcation, the best I could do was to get that there had to be parallel edges. It's co-migration, polyclonal seeding. So I'd say, oh my gosh, let me publish a Nature paper that metastasis is polyclonal seeded because I saw my data. But, just a little tweak, if you're just aware of some ambiguity in the tree, suddenly it's back to our old friend, monoclonal seeding. Nothing interesting to see here. The usual stuff from the 60s that we already knew. Okay, so I have to be careful. So let's look at this a little bit. In the other example I showed you, this was this sort of beautifully illustrated tree with this multiclonal seeding. To show it in a more mathematical way, here's the migration graph, primary breast tumor, seeding these different things. Look at the craziness. The lung receives cells from both the rib and the primary breast tumor. Multiple migrations between rib and kidney, et cetera. 14 migrations, polyclonal or multiclonal. So if you sort of look at the ambiguity in the clone tree as you're doing the migrations, if you just change things ever so slightly, equally consistent with the data is a pattern like this, where from the breast tumor, cells migrate first to the lung, and then from the lung seed all the other metastases. It's actually very consistent with what we know about the patterns of metastasis, where the lung is very well connected to the circulatory and lymphatic system, so seeding to the lung first and then going on. It's very common. Okay, so that's sort of the end, and I've maybe talked too long already. So, the notion here is that if you're going to say that migrations are very complicated, you should first rule out the simple explanation, right? simple monoclonal migration is like the null hypothesis. You got to have pretty strong evidence to reject the null. So how do you do that? You need models to actually show that. So this is one of them. This thing was called mocking. I forgot to say that, right? It's an acronym for something that I forget. M is migration or metastasis. I don't remember. And then, you know, this is all sort of a prerequisite to really doing the interesting biology, which is actually trying to figure out across different patients, what are the drivers of metastasis? What are the biases in migrations? We really can't do that until we have a good model to actually explore the different migration patterns. So that's sort of what we've started with, and this idea of the simultaneous inference. So just to kind of bring it back to the analogy at the beginning, I think, you know, we should all be sort of thinking about, you know, don't make a mess of a novel data set by using tools improperly, right? And instead of slicing our cake this way, we really want to have this a stainless steel cake knife, very fine grained, with an accompanying stainless steel cake server. And even in this multi-layer cake, you can get a very beautiful slice. So very beautiful analysis with the appropriately engineered tool for the job, right? I wouldn't want to cut wood with these tools, but I do want to cut a cake with them. So there we are, and that's the acknowledgments. And yeah, I'm always recruiting, so if you're looking for something new to do, talk to me. And as I said, I'm here and happy to talk to any of you throughout the next couple of days. So thanks.
I have more of a maybe not scientific question. I understand why we study evolution, because it's our history. We
Sure.
want to know what happened. Why? What will we like? Every patient has
Yeah.
his own history. So
Sure.
will it help the patient once we infer what happened with his tumor, or is it just abstract interest? What happens with cancer?
Right. So, you know, after the fact, of course, once someone comes into the clinic and they have 10 metastases, then maybe, you know, building that tree might not help that one particular patient. However, if we sort of have a sense of, does metastasis happen early in the evolution of a tumor, or does it happen late? If we have a sense of what exactly can we infer, because often we don't see it till late, could we infer what mutations or epigenetic states or what have you, the cells that can metastasize have that distinguishes them from the primary tumor? Are there such things? If we could find those and we knew them and we could somehow drug them, then perhaps we could think of preventing metastasis. Surgically, you know, there has been this discussion of how exactly should you surgically treat a patient. Do you cut out the lymph nodes always? And if you think that metastasis always goes through the lymph nodes, then often when they'll do surgeries, standard of care is to cut out not only the primary tumor, but surrounding lymph nodes. Cutting out lymph nodes is not always a good idea. They're real connected to the immune system. It's very, you know, so if we know that that's not really going to help because metastasis will still find a way, then that might change how we also do surgery. So all of those things are actually starting to happen, you know, and you know, again, it's sort of secondary effects from understanding the evolutionary process. But the tree itself is sort of, you know, a structure that helps us do these other things. Yeah. Hi.
Hi. So since we're not in the 70s and the 80s anymore, what are the prospects for having really large data sets of many examples of this kind, and then maybe doing deep learning on them?
Yeah. So I won't answer the second part about the process or deep learning, but the prospects for bigger data sets are actually, you know, quite good. You know, because DNA sequencing has gotten cheaper, because you can sequence from smaller biopsies, there are, you know, increasingly numbers of data sets where we have matched primary and met from the same tumor, or we have a primary that we've sequenced when the patient initially came in and the patient relapsed or metastasizes and we get the mets later, or even we've done a treatment where there's intervention. So these data sets are increasingly being generated. I'm sure, I don't want to project numbers, but they'll easily think of like TCGA with tens of thousands of primary tumors. We'll get to that with matched primaries and mets in, you know, the coming years.
So and when you have much larger data sets, you could conceivably actually search over the metaspace.
Yeah.
What the
Yeah.
optimization function is in search of.
That's right.
Of clearly differentiating.
Yeah, how do we parameterize them? Can we learn the biases between, a certain, it's clear that certain tumors have preferences for where they metastasize. And obviously some of that's local. The colorectal tumors go to the liver. But there are other biases that are sort of more strange, like breast tumors tend to go to the brain. And, is that just because of the connectivity of the circulatory system, or is that because inherently those tumors are better evolved to be in these different tissues? So these are real fascinating questions that with enough data we could start to kind of learn these parameters.
I have a couple of questions. First one, when you do the metastasis, you mentioned that you are not necessarily looking for driver mutation or percentage.
Not at this stage, no, yeah.
So my question was relating to that, where you see are the drivers of driver mutation even distributed in the different locations?
Right. So when people have looked for mutations that are, say, recurrent in metastases that aren't in the primary. So you could do a study, and people have done studies like this, where maybe you sequence 100 patients, their primary breast tumor and their Mets. And then you try to find mutations that are shared among the Mets of these individuals that weren't present in the primary tumors of these individuals. And that would indicate that these might be recurrent mutations that are somehow promoting metastases. The literature at the moment is very unclear and contradictory about whether there are such mutations that drive metastases. In some tumor types, in small sample sets, people find recurrent mutations and suggest that they're drivers. And then there are other papers in different tumor types, or sometimes the same tumor type with different patients, and they'll claim they don't find them. So it's still kind of early days for knowing whether metastasis is driven by mutation, driven by non-genetic events, totally sort of random. Any primary tumor cell could equally well metastasize. There's nothing inherent once you're a tumor cell. All these are still open questions.
So another question. You said that homoplasia was, I think, not justified in a way. My question is, if you look on the mutation that happened, is it possible that one mutation can be correlated with another mutation?
Yeah, I mean, there are all sorts of, strange things that can happen where you can have, mutations that seemingly correct for defects in other mutations. There are examples like this. There's, of course, no homoplasia is a good starting assumption, but you have to be careful in using it. Maybe it's a reasonably good assumption for single nucleotide changes, but when you start deleting segments of the genome, this is another way that mutations will appear and disappear. So there might be a mutation, a single nucleotide, then later that segment of the genome is deleted. So the mutation disappears. And these interactions seem to be happening. They're definitely happening a lot, whether they're random or whether they're selected for. Again, still, I would say early days in knowing this. But there are definitely going to be interactions between these mutations in certain tumors. It's just a question of how often is that important.
Next question.
Yeah, we should.
So you spoke about the process of cell lineage, okay, which is definitely a tree,
Yeah.
correct? And then you have this network, but the network is over several cells.
That's right.
correct?
Yeah.
So how so you cannot have a network over one cell. So how
That's right. Yeah. Well, so the reason why we sort of draw this graph and we talk about it in terms of the network is the nodes of that graph suddenly became the samples again, not the cells, right? And part of the reason for us doing it that way is that's really consistent with what the literature talks about when they're saying that metastasis is a monoclonal event or a polyclonal event, or they're reseeding. All of that language is about what's happening to individual cells between two sites. And so the right language for talking about all of those events, it's different cells, it's not the same cell that went back and forth, but all of that language